Hey, I wanted to make a video of me going over the cell structure, and it's going to be eukaryote and prokaryote functions. This is a cute little quote by Anton van Leeuwenhoek, one of the first creators of the microscope, about how he looked at his stool under a microscope, and he explained what he saw in his stool, in his poop that he looked at, and so he could see what we now probably think of was a protozoa called Giardia, but that's just really cute. So I did want to review a couple of things about the microscope. So remember that when we use a microscope, we are magnifying the size of something. So we're looking at, for example, bacteria or the letter E or whatever under the microscope. We're magnifying it, and you get the total magnification by multiplying the ocular times that objective lens. Remember, scanning was 4, low power 10, high power 40. And so, but the resolution is your ability to see fine detail. And so it's the opposite of blurry. If you take a 4x6 picture and you try to blow it up to a 16x20 picture, you're going to lose resolution, and the image is not going to be clear. It's going to be blurry. It'll be distorted. Contrast is the difference in shading. We generally will stain objects like bacteria or yeast so that we can see them. The field of view, that's the circle that you're looking in. And as you increase your magnification, the field of view actually gets smaller. As you increase magnification, that's what you see in the circle that you're looking at gets smaller. Depth of focus, this is the thickness or vertical distance of a specimen that can be seen. So this is like if you have ever done the lab where you look at the colored thread, you can tell like which thread is on top, the thickness or vertical distance of a specimen that can be seen all in sharp focus at one time. And then of course the working distance is the distance between that objective, that magnification objective, and the actual slide. Okay, so let's go into the cell theory. So this is 3 things based on highlight who said it. Schleiden, Schwann, and Virchow said three things about cells. All organisms are composed of cells. So every living thing is composed of cells. Cells are the basic unit of structure and function. And so all living things are made up of cells. Cells are the smallest living thing. And then cells come from pre-existing cells. And I always want to kind of focus on that third one, but know who said all this. But and you may, if you had printed out the student notes, you may need to write in who said it. And so let me get my pen here. You know, write in the scientist that came up with that. But on number 3, it says cells only come from pre-existing cells. Today in 2025 or 10,000 years ago when they were studying cells, I don't know if they were studying cells then, but whatever. Cells only come from cells. Like, you know, back a long time ago they thought this sounds so silly, when it rained that frogs appeared. And frogs don't just appear, that's ridiculous. Same as that cells don't just appear. That's ridiculous, right? And so I always want to point that out because I find that very interesting that they all agree that cells don't just appear. Cell size, cells are very small so that they can get materials in and out. I always think about, like, if you're going to cook, for example, chicken, little tiny small chicken, like cut up, will cook much faster than a big piece of chicken if you're trying to grill it or cook it on a stove. The little pieces can get the heat in and out faster and cook throughout. You know, can cook, if you take one big piece of chicken and cook it on a grill, and then you take that same piece of chicken and you chop it up into small pieces, the small pieces are going to cook faster. Even though the total amount of chicken is the same, it's because cells are small because they need a large surface area to volume ratio so they can get particles in and out. So that's talking about the surface area to volume ratio. Okay, you already know this. I know you do. There's 2 types of cells. There's prokaryotes, cells that don't have a nucleus, and they evolved first. And they do not have a nucleus. Highlight, underline that. And then there's, it says prokaryote cells are very common in the air, in our bodies. This is right in the word. This is bacteria. Another word for prokaryote is bacteria, and they do not have a nucleus. Write that in. Prokaryotes do not have a nucleus or any of the other organelles associated with that. Eukaryotes, which is the next topic, well, next in a little bit, they do have a nucleus. Okay, so but these are bacteria. The word prokaryote and the word bacteria, you know, kind of know that prokaryotes are bacteria. We talk about the domain bacteria versus the domain archaic. So the domain is, some people will say eubacteria or just bacteria. They do cause serious illnesses like tuberculosis. That's the #1 bacterial infection that kills the most people in the whole world. Not necessarily in America, we have medication, but worldwide, in places where there's great poverty, lots of people die from a bacterial infection in their lungs called tuberculosis, and then other infections, like it says, throat infections. You know, so a lot of different types of bacterial infections are in the domain bacteria. They also decompose the remains of like dead leaves and trees and animals, and then we actually have bacteria in our intestines, in our abdomen, that help us digest food and even digest medications that we take. And so, but your book will tell you that most bacteria are actually good. They're actually good. Now, the domain archaic, they're different. Now, both of these don't have a nucleus. That's why on the left-hand side you can see where it says prokaryotic cells. Archaea and bacteria don't have a nucleus. But the archaic are, by definition, they live in extreme environments. Salty, hot, they're unique. This is not what we come in contact with every day. This is not like regular bacteria. Bacteria come in one of 3 basic shapes: the bacillus or rod shape that you see in the picture, coccus or round, and then Spirillea or spirochete is the curly ones, the blue ones that you see in the image there. So highlight that. You've got coccus, which is the round, bacillus, which are the rod ones. They look like a sprinkle, right? And then the all different shaped curly ones we call Spirillea. In bacteria, they have a cell membrane, and the whole next lecture is on cell membranes. But you can see that they're made of phospholipids, so phosphate head, saturated fat, and an unsaturated fat. Head is polar, tails are nonpolar. It's got 2 layers, so here's one and here's one. So phospholipid bilayer. We call it, this says cell envelope, but it could say cell membrane, plasma membrane. That outer layer, it says, has plasma membrane, cell wall, and some have a sticky glycocalyx layer. The plasma membrane is made up of phospholipids and proteins. For bacteria, there's a cell wall highlight made of peptidoglycan, and then some bacteria will have a thick, sticky substance on the outside. I want you to think of caramel, thick, sticky substance on the outside of the bacteria, and this really helps the bacteria stick or adhere to surfaces. Think about, or the best example I could give you is the bacteria will stick to your teeth, the slippery enamel on your teeth, and the bacteria will stick to the teeth, and then the bacteria will all stick together because of this glycocalyx layer. Caramel, think caramel. And then we have to go to the dentist every 6 months and get that plaque, we call it, but it's bacteria stuck together because of glycocalyx literally plucked off or scraped off. Yikes. Okay, here's a picture. All of the liquid inside is cytoplasm. So you look at your notes, I'll look at this picture, but all of the liquid inside is cytoplasm. And then there is a cell wall, so that's this part right here. The cell wall is red. Okay, the cell wall is red right here. and that's made of peptidoglycan. So this is all the cell wall, and then that thick, sticky outer surface is the glycocalyx layer, right? A capsule, or, you know, kind of like I gave you the example of caramel. Okay, then there's also DNA. This, I'm just going to point to this picture because I like to be visual, but this DNA right here is just in the middle. We call it the nucleoid region. It's just DNA in the middle in the form of, you know, loosely coiled DNA right there. Bacteria, this is all bacteria, prokaryote bacteria. They do have ribosomes. That's where proteins are made. Proteins are made, or proteins are synthesized. Now let's look at these little hairs. This long hair down at the bottom is called a flagellum or flagella, and that's used for movement. That's used for movement. And then all these little hairs are called fimbriae. That's used for attachment. Circle that on your notes. Used for attachment. So flagella was used for movement, and fimbriae is used for attachment. An example, the bacteria Neisseria gonorrhoeae that causes the sexually transmitted infection gonorrhea uses fimbriae to attach to the mucosal lining in the vaginal canal, causing the sexually transmitted infection. So the bacteria can stick in that area because of fimbriae. And I think of fimbriae like Velcro. It's hairy, but it's used for attachment. Okay, let's look at this in the notes. The DNA is found in the middle in the nucleoid. Oh, there's additional DNA, little tiny circles of DNA called plasmids. So this is, you can see it's red like the regular DNA. Extra DNA, extra chromosomal DNA. Ribosomes, highlight, underline, circle, that's where proteins are made. And it says inclusion bodies store substances such as nutrients inside the cell. Okay, the bacteria have flagella for movement, propels the bacteria. Maybe write in the word movement there. Fimbriae is used for attachment. And then we didn't cover this one already, but there's this right here. This is where it says it. There's a thicker tube-like. Bacteria can extend a tube-like structure to pass DNA. And so I always kind of draw it out like this. Here's a bacteria. Here's another bacteria. Only some bacteria can extend a tube-like structure, and it can exchange DNA through the conjugation pili, or the picture says sex pili. Cilia are also hair-like extensions that are more numerous, but they're not generally found on bacteria. And so we have long for movement is flagella, and then short hair-like extensions, fimbriae, are for attachment. Okay, some bacteria do photosynthesis like plants. How cool is that? I know there's algae, and that's not bacteria. Algae does photosynthesis, but right now we're talking about bacteria that do photosynthesis called cyanobacteria. This says that most bacteria are actually heterotrophs. They don't do photosynthesis. But not all. The ones that do photosynthesis we call cyanobacteria, and they have thylakoids. A thylakoid is an organelle. We're going to talk a lot about it when we get to the plant cell, but a thylakoid is an organelle found in plant cells. I mean, it's inside the chloroplast. It's stacks of grana right here. The whole stack is called a thylakoid, and it's for photosynthesis. So some bacteria do photosynthesis. We call them cyanobacteria, and they have inside them chloroplast, and that's not on the slide. You can write it in, or you can just know it when we get to plants. But plants do photosynthesis, the process of making sugar. So do these bacteria in an organelle called the chloroplast. And the chloroplast has little, I think of them like cookies or coins stacked up, called thylakoids. So there they are stacked up, and the whole stack is called grana or granum. Okay, so there's this theory that it's like, how in the world could we have gotten a eukaryotic cell? That's what this is coming from. How in the world? And so the theory is that one larger cell engulfed a smaller cell, and then now there's like a small cell inside a large cell, and that could have evolved into organelles. That's just a theory. It's called the endosymbiotic theory. And why do they think that? Well, your cells have the DNA inside them. Your mitochondria and also the chloroplast have different DNA. And the mitochondria and the chloroplast both have like a phospholipid bilayer around them, like almost like they're their own separate little cell. And so mitochondria and chloroplasts both have their own DNA. They both have cell membrane, a double membrane. So they think, well, maybe that's how this happened. But this is a theory. It's just a theory. You can see where one large cell engulfed a smaller cell, and that could have evolved into the first eukaryotic cell. Okay, so few eukaryotic, there's different ones. There's plants, animals, fungi, and protists. We talked about that already in the course. There's animals, plants, fungi, and protists. They're all eukaryotic cell, which means they have a nucleus and organelles. They have a nucleus and organelles. Each organelle has a separate function. The first one is the nucleus, and it has the DNA. So if you look here, here's the nucleus, this right here. And it has inside it the DNA. Also, the nucleus has an outer membrane right here called the nuclear envelope. It has nuclear pores. Inside the nucleus is also the nucleolus, and it stores the RNA and proteins, RNA and proteins. Ribosomes are formed here, and they migrate out. And so these are ribosomes out here. They also, that's where protein synthesis occurs. Let's see, right here, it's labeled right here, free ribosome. Okay, right outside the nucleus, there's a plant cell. Is the endoplasmic reticulum. If it's got ribosomes attached, you see the ribosomes? It's rough. If it doesn't have ribosomes attached, it's smooth. So go back and look at this picture. These right here, this is the endoplasmic reticulum. That's rough. It has ribosomes attached. It's rough. The smooth has, it doesn't have ribosomes attached. That's where lipids are made, so it's shiny and smooth. It functions to produce lipids. The rough ER functions to produce, functions to transport the proteins. Okay, it tells you there, the rough and the smooth ER. The Golgi is where processing, packaging, and secreting proteins. You're going to stick a carbohydrate on the protein and then transport it to the cell or out of the cell. Lysosomes, you're going to remember this because Lysol, it cleans up the cell. Functions in digesting material made in the cell. Highlight that. Oh no. Maybe I didn't click on it. There we go. Okay, and then when you go back to the smooth and rough ER, make sure you have written in right here. Rough ER transports proteins. And then the smooth ER functions to produce lipids. Okay. So here you can see a picture of some of the organelles. So pause this video and look, you can see the vesicles coming in and out. Peroxisomes are similar to lysosomes. They actually produce hydrogen peroxide, builds lipids and breaks down lipids and ethanol. Okay, so highlight here, right, the hydrogen peroxide found in peroxisomes. The vacuole is a large storage molecule. So it's large in plant cells to store water, but in animal cells it'll store fats. Plant cells have large central vacuole, which may take up to 90% of the whole cell. When you see that, look at this plant picture right here. See, this big thing is, let's see, right here. See, this whole big thing is the vacuole for storage. It stores water. Okay, this is the chloroplast. The chloroplast is where photosynthesis occurs, the process of using photolight to make sugar. Plants, some bacteria, cyanobacteria, but plants will have a chloroplast in them, and algae too. Plants, algae, and some bacteria possess their own DNA, their own double membrane. That's that endosymbiosis thing. Okay, so here's the chloroplast, has a double membrane, and then it has these thylakoids stacked up, right? And it tells you the granum is enclosed compartments of stacked thylakoids. Thylakoids are disc-like surface, possess chlorophyll, that's the pigment green. And then this jelly-like substance over here is the stroma, all this extra. Here's a picture close up. I know my picture was better. That's what you're thinking. Mitochondria, to me it looks like a hot dog with mustard. It does have its own DNA. That's not that mustard thing. The mustard is called the cristae. This is where ATP is made. The sugar made in photosynthesis or eaten in our food is broken down, and ATP is produced. One thing I think students sometimes find confusing, plants have a mitochondria. They do. So plants will make their own sugar with photosynthesis, and then they will break that sugar down with the mitochondria. So mitochondria break down carbs to produce ATP. That process is cellular respiration. We're going to study that really in depth in another chapter in the class. They have their own DNA. They have a double membrane found in nearly all eukaryotes, including plants. Bound, and they have this like folded cristae. There it is. Okay, so let's pause the video real quick, and if you haven't already, let's draw this out. Okay, so there's your cell. Remember, that's made up of phospholipids, right? And I'll spare you, but it's got the phosphate, the lipids. It's got two layers, right? It's got phospholipids and proteins. I won't draw this all the way around, I promise. Phospholipid. Okay, so just kind of showing you, that goes all the way around the cell. That's the phospholipid bilayer or plasma membrane. Okay, every cell has in it a nucleus. So draw this with me if you haven't already. The nucleus is where the DNA is stored in the form of chromatin. Okay, side note, DNA has two forms. It has chromatin, loosely coiled DNA, and then it has tightly coiled DNA in the shape of an X. We call it a chromosome. So this is chromatin, and this is chromosome. Okay, so cells have a nucleus in the form, and it has the DNA inside it. Cells also have inside the nucleus a nucleolus. Okay, the nucleolus is where RNA and proteins are. Okay, so and the nucleus has a nuclear membrane, right? You'll learn about that. Okay, now, can I change colors? Maybe. Let's see. Oh yeah, now I'm working. Okay, just outside the nucleus is the endoplasmic reticulum. And if it's got ribosomes attached, it's rough. Okay, remember that you write it out, but I won't because I know it makes a loud noise. Endoplasmic reticulum. There's rough and smooth. Rough has proteins attached, has ribosomes attached. Rough has ribosomes attached. It's for protein transport. Write that in. Smooth is where lipids are made. Okay, so that's your ER. And usually the smooth is just outside from the rough. So it's rough, just outside the nucleus, and then the smooth. Okay, keep on going out of the cell. You have the ribosome. That's where, that wasn't very good, was it? Let me try again. I can do better. It looks like a hamburger bun. But it's going to be way smaller than this in relationship to the nucleus. But I made it big so I can kind of show you its function. Ribosome, that's where, write it in on your paper. That's where proteins are made. Remember, proteins are made up of amino acids. Okay, you draw this in and you write the function in too. Okay, you have I feel like the mitochondria should just be red, shouldn't it? You have the mitochondria, looks like a hot dog with cristae. It's got its own DNA, its folded membrane. That's the mitochondria. That's where ATP is made. That's where cellular respiration happens. ATP is energy. ATP is made in the mitochondria. It breaks down carbs to produce ATP. That's called cellular respiration. It's in both plants and animals. So maybe write in right there, cellular respiration. The nickname, oh my goodness. There we go. The powerhouse of the cell. Okay, you have also, we'll make it blue because that's like a nice clean color. Lysosomes clean the cell. There's a bunch of them, but I'm only going to draw one. There's a bunch of ribosomes too, but I only drew one. Some animal cells may have a vacuole for storing fat. And then if this was a plant cell, it would have a chloroplast. But it's round, so it's probably not a plant cell. Okay, listen to me. Let me help you with this. Okay, remember that the function of the nucleus, that's where the DNA is. What does DNA do? DNA is the instructions to make proteins. Okay, I always give this example in class. If the DNA, right here is the DNA, is the instructions on how to make a protein, think about it being like a recipe of how to make, turkey and dressing on Thanksgiving. How to make the dressing, okay? It's in the whole cookbook. So you have a whole cookbook of recipes. That's your DNA. The entire cookbook makes everything. But I want one gene is like just the instructions to make one protein. So just the instructions to make the turkey and dressing, or just the dressing. Okay, so we're going to take, I'm not going to take the whole cookbook out there. I'm just going to take a picture of the one page I need. And that's like RNA, which is where it's in the nucleus, but nucleolus. But the DNA is going to unzip and send the mRNA, that's messenger RNA, out of the nucleus. That's like the photo of just that one page in the cookbook. Okay, so remember that DNA is like a cookbook. It has all the recipes to make protein. Okay, when you make a photocopy of just one, that's like mRNA. It's different. So photo is A pairs with U, not A pairs with T. But it's similar. Okay, and so it goes out and it's going to be transported all the way over here to the ribosome. The mRNA is going to go through the ribosome, and the ribosome is going to read the mRNA, and it's going to put together, it's actually going to put together the proteins. The proteins are made up of amino acids. So the amino acids that we've eaten, The ribosome is going to put them together in the right order. It's going to read the mRNA and make it. Well, then that amino acid chain right here that was just made has got to be folded. Remember, primary structure, secondary structure, tertiary structure, quaternary structure. It will go out into the, I'm out of colors. There we go. It'll go out into the Golgi. I didn't go over that one. The amino acid will go over to the Golgi where it is stamped with a carbohydrate, and the Golgi will either transport it out of the cell or back into the cell. So the Golgi apparatus, it tells you in your notes, processing, functioning, and secreting proteins and lipids. But think about packaging. It puts a stamp on it, a carbohydrate stamp, and says you go out of the cell or you go back into the cell. Okay, so you look at this picture, and also at the same time, I want you to look at your notes, and let's just go through it real quick. This is a eukaryotic cell with organelles. You have the nucleus that has the DNA. It's chromatin, loosely coiled DNA. The nucleolus has inside it RNA and proteins, and the nucleus has a nuclear envelope around it. Ribosomes is where protein synthesis occurs. Endoplasmic reticulum, it says complex system of membranes, channels, and sacs. There's rough ER that transports proteins, and there's smooth ER where lipids are made. The Golgi apparatus, hopefully you highlighted, packaging and secreting proteins. The lysosomes clean all this up, digesting material. Peroxisomes also do. And then also the central vacuole cleans it all up too. Chloroplast is where photosynthesis occurs. The mitochondria is where ATP is made. Okay, then we have also the cytoskeleton, and that's going to be actin, intermediate, and microtubules. Okay, this is outside. It says cytoskeleton is made of proteins, components that run throughout the cytoplasm. So this is going to be inside the cell. Inside the cell, it's like a skeleton, literally cytoskeleton. And there's actin, T-I-N, which is the thinnest one, intermediate, and then microtubules. I want you to think about like this one being a thread, intermediate being more like a toothpick, and then this one is more like a straw. Okay, so thicker. Intermediate is in the middle of actin, T-I-N, and microtubules. That these are protein that run through the cell. You never see them on drawings because they would mess up the cell. They would mess up the picture. But, you know, so if here's your cell that we just drew out, the cytoskeleton is like all over trying to hold its structure together. So there's pictures, actin, intermediate, and microtubules. Okay, centrioles, when we talk about cells dividing, the centrioles make spindle fibers that move the chromosomes around. They're shortened cylinders that direct the movement of microtubules during mitosis. These are for movement. All of these, these are moving chromosomes inside the cell. Of course, cilia and flagella are hair-like projections. Highlight, cilia are short and numerous. Flagella are long and they spin like the propeller of a boat. There's only usually a few flagella in comparison to the cilia. So here's a picture of a paramecium. It has a bunch of little cilia all around it. This is a flagella, like the bacteria E. coli. It has flagella all over it. Okay, I hope that was helpful.